So one of the things that I like to do with these meetings is I like, I like a lot, I like a lot of improvisation and I like a lot of repetition because I think for lay audiences, it's really important to hear the same concepts many times during the course of the day, and then it will set in. I'm a former middle school teacher, so that's why I think that way. So I'd like to start out today talking about CAR T. Because that's what's on top of everybody's mind. But there are obstacles with CAR T. So I'll start with a real simple one. Who should be excited about CAR T? And we can start with anybody, and I hope that you will all add and pass the mic along.
Since I have the microphone in my hand and it's a simple question, I will take that one. No, I mean, we were very excited when it came, because as all drugs and all treatments that we use, all new ones are tested in patients who have very advanced disease. And because there it sounds mean, but oftentimes you have nothing to lose, right? It's worth the try to find something that still works if everything else has failed. And usually we see response rates that are, well, okay, there's some patients have a benefit, most don't. And in the CAR T cells, it looked like it's almost the opposite. Most patients had a benefit in those very late lines. What we have learned now, as with all other treatments and drugs that we have as well, the earlier we use, it seems to be the better. And we're still in early stages, and obviously here are people who are much more proficient in this area. But I think one exciting thing is that it even works if the disease is really not responding to a lot of other things anymore. That's one thing that patients who are basically didn't have a lot of hope have hope again. And another is that we might use it earlier, which then benefits everyone because we can use it in areas where the patients are still in much better condition. They feel better. They have had less side effects over the course of their treatments. And I think those are the areas. And
I am not the moderator of these things normally, and I just Jenny was very ill and did end up leaving earlier. And the other thing I didn't do, which I realize, is I didn't introduce our fourth speaker, who is Dr. Irving from Memorial Sloan Kettering, and she'll be speaking this afternoon. So I asked her to join us for this discussion. I would never make it on the Oprah show because I don't understand protocol. So I will pass that back to you now. Thank you. Does anybody have to add to that? So who should be excited about CAR T?
My turn?
Yeah.
All right. You put up the slides. If you want, I can go if while you're waiting.
Just to add, I think, when we ask who should be excited, just now I see patients with newly diagnosed myeloma or early stage getting excited about CAR T, but I'd say pause on that. Only patients with 4 or more lines of therapy, it's approved. And, CAR T, while it does have great response rates, there are situations in which it doesn't work. It takes time, to prep the patient for it. And there are other good therapies. So we have to do it in everything in a sequence, and the CAR T is not the right thing for people initially, and it's only in certain situations later. So while it's exciting, it's not for everyone and not in the beginning. And I think that's quite important for patients to know, because many times patients hear about this like miracle treatment, but it's not miracle for everyone, and we have to find the right patients that it works for. It also takes some time to make the CAR T cells. So if the disease is very active, then that's not a good patient also to try to do CAR T cells for.
Can we get the slides for?
Thank you. What, you're not talking? I can't believe it. You have nothing to say? Come on. It's like a first. I can just put in, careful what you wish for, because CAR T, at least in lymphoma, we have a longer track record. And I have some patients who are 4+ years out who still get GCSF twice a week because they don't have good blood counts. Now that's a CD19 directed CAR, that's against B cells. But why should your knocking off your B cells influence your neutrophils? We don't understand that. They also this person, she's got a low platelet count. And yet she's got a cellular marrow. Her platelets are in the 30s. So when she had to have her hip replaced, she had to be tanked up with platelets. She's grateful. Her lymphoma is gone. We're not quite seeing a long enough term follow-up with myeloma patients, so we don't know. Plus, as you know, the waiting list is ridiculous. So I've had patients die while waiting to get a CAR T-cell therapy. It's horrible. And so we struggle with that. Both BMS and Janssen are struggling with manufacturing. So it's CAR T 101 right now. And so we really have a ways to go.
What are some of the obstacles that you're talking about? Can you explain those in greater detail?
Waiting
So like,
list.
what are the rules that you have to adhere to?
Waiting
Who can get them?
list. Waiting list.
So how many CAR T procedures are there done generally in a month in this country?
I have no clue, but it's not a lot. I mean, we have how many?
What do you mean? How many patients? A waiting list, we have a long wait list
Yeah,
and
we
we
have at
would
least
like to
10
wait
patients.
at least.
And our next slot is November. So that's a huge problem. I mean, we're just opening Janssen product and we're going to be sharing it with the University of Rochester because they don't have it either. So, and it's just that the manufacturing has taken forever. Mostly because I don't think companies realize this isn't a drug, it's a living thing. And when it's a living thing, people are variable. So somebody's immune system's beat up, you may not get good lymphocytes out. And so right now we're trying to understand why that is. The Penn group had a really interesting study where they grabbed CAR T cells like right away. The patient had high risk features, and they found that the type of lymphocytes they got then were much better than you waited for a patient to relapse refractory to 4 prior lines of therapy, including an IMiD, a proteasome inhibitor, and a monoclonal antibody to CD38. Those are patients whose immune system is fairly beat up. So now the whole issue is, well, maybe we should store lymphocytes on somebody who's high risk. So therefore, you've got a much better set of lymphocytes to manufacture. However, some of the manufacturing can only be done on live, fresh cells as opposed to frozen. So there's a lot we have to learn to make this better and also understand better so that we don't treat everybody with a CAR T cell.
And that brings us to the alternatives to CAR T and bispecifics. How do you see bispecifics coexisting with stem cell transplant, coexisting with CAR T?
Yeah, I think that the reality of CAR T in its current iteration today doesn't live up to the hype. There are real issues with supply chain as well as technical expertise and employment levels at the manufacturing facilities. The bispecifics, of which the first has already been approved in the European Union and anticipate approval in the United States, will be an off-the-shelf, something that'll be amenable to repeated administration as opposed to CAR T, which is a one-time and done. And given the very high response rate with bispecifics, I think it will be very, very powerful competition for CAR T, where I would anticipate no wait lists for bispecific antibodies and the ability to continue treatment long-term, which is not generally the case for CAR T. So I think it's actually more exciting than CAR T. Nothing's perfect, and the bispecific antibodies have been associated with some extremely, extremely serious infections. And that's part of the learning curve, unfortunately, is to just understand what it does to the normal healthy immune system and what can be done to prevent these exotic infections. This business with CAR T, you know, everybody talks about effectiveness, but some of the side effects can be quite sobering. With one of the products, there have been reports of late, after 6 months, Parkinson's disease. I've never met a patient who's been told about the side effect. It definitely occurs, and regrettably, CAR T has a mortality rate. I mean, people don't always survive CAR T treatment, and I would say that mortality rate, at least at our institution, is between 5 and 10 times the mortality rate of autologous stem cell transplantation. I think the exciting thing, Greg asks about, what about getting excited about CAR T? I think that Professor McCarthy was correct when he said it's CAR T 101, and that in its current iteration, there's some serious obstacles, and it's not that great, but the opportunities for further development, different targets, multiple targets, CAR, not T, but CAR with other cells, suggest that there's a very high research upside to try and develop better products than what we currently have, which are highly effective, but not the magic bullet yet.
Will the patients in this room ever see the day where stem cell transplants become obsolete?
Maybe.
Why maybe? What is the thing that makes it a maybe?
Well, it still works. And it depends. We're in the US and we're very lucky. We have access to a whole bunch of things that people outside the US don't have. So even in Europe, there are high-dose malformed stem cell rescue is part of the treatment continuum for a lot of patients. And a lot of other drugs are not available yet and won't be for a while. And that's just because of the way medicine works in other countries. I'm not going to say our system is great, it's chaotic. There's a whole bunch of things we could spend an hour on what's wrong with healthcare here, but we do have great access to drugs. It's just all financially unsustainable. That's another issue. But yeah, I do think, and then high-dose malformed stem cell rescue is fairly cost effective as opposed to a quad, which is going to be what we're doing next. We're going to go to a 4-drug regimen. And so I do see the day that potentially will go away, but we also have to think of what can we do to replace it? Will it be a CAR T cell? But we may want to collect stem cells so that we can rescue people who have pancytopenia after the CAR T cell therapy, which occurs. So there's a lot we don't understand. And as was mentioned earlier, Janssen is studying it. We already know that ide-cel will likely be moved up because one of their trials looking at salvage, earlier salvage with dealer's choice versus CAR T is going to be, it's a positive study. So we'll be using it earlier, but then if somebody has beat up cells and you can't get any drugs into them because they have low-grade pancytopenia, we better collect everybody up front. So we'll still be collecting stem cells. It's just that we may not be using them with high-dose malformed. We'll be using them with something else.
So stem cell transplant is 40 years and running in multiple myeloma, and to date hasn't been knocked out of that spot. Most investigators report a fraction of patients who are 10 and 20-year survivors after stem cell transplant without any maintenance treatment. And it works a completely different way than all of the other available treatments. As things go along, the biggest concern that I have in my practice is the fact that patients who get stem cell transplant do have a higher risk of second cancers. And as people live longer and longer and longer, they have time to develop these problems. And when they develop, they're quite unpleasant. And so it's, for me, kind of the big problem in the back of my mind about, will transplant lead an otherwise long-term good outcome patient to develop a second cancer? But for right now, I think it would take quite an impressive outcome to dislodge stem cell transplant from our current practice.
Yeah, just as a follow, we've had patients who didn't get a transplant, but then got lenalidomide-based RBD and have developed acute lymphoblastic leukemia. We don't understand that. We have no clue how that occurs and why it occurs in a fraction of patients. We also don't understand. We also don't understand there's a paper from Olangren, about 10, 15 years ago, looking at the Swedish Cancer Registry. And what he showed is in that patients who had MGUS, monoclonal gammopathy of undetermined significance, Dr. Kyle's observation, and smoldering myeloma, who hadn't been treated yet, still had a 2 to 3-fold increased risk of getting AML, acute myeloid leukemia, and MDS without therapy. So myeloma itself predisposes people to developing hematologic disorders. We just don't understand how this works. Then, of course, you pile on drugs on top of it, the risk starts to go up. But as to Dr. Gertz's point, it's better to be alive to be at risk for developing a second cancer than not to be alive, and you won't get a second cancer. So we just need to do a better job of identifying those patients who we should treat differently because they've got a different genomic makeup than other patients who don't develop the second cancers. Oh, and by the way, there is are you 19 or 20 years from your stem cell transplant in the audience? Where are you? Well, anyways, there is one of our patients is here who has transplant about 20 years ago. No maintenance, doing well. And I don't understand what's different about him relative to other patients. So we need to understand that a lot more.
Two weeks ago today, we had a roundtable in Hamburg, Germany, and we had a session with a panelist who was the first CAR T patient that this doctor had treated, I guess 3.5, 4 years ago. Hasn't been on any treatment since, and he's just doing fine. Not that he's the typical case, but there are cases like that out there, and it's really exciting to see them. And he had been through 10 lines of therapy, and has been without therapy now for going on to 4 years, and is feeling just wonderful. What do you see as the future? I mean, yeah, first of all, I guess we have 2 approved CAR T cell products right now. What do they presage for the future? I mean, one looked at one target, one goes after 2 targets. What is the future looking like for CAR T? How is it going to change? Is it going to stick with figuring out how to exploit BCMA, or is it going to be other things that are going to be coming up that are in the research right now?
So we already have other targets being looked at, right? So the GPRC5D CAR T cell is there, and similarly, you have a GPRC5D bispecific. So we have that trial open at our center with the CAR T cell for MSK, but there's a multi-center study also ongoing with that. So we already have that target. Then the next one is cevostamab or FCRH5. So there are different targets being looked at. I don't know what's going to be next, but there are many different targets being looked at. So I think, how do we combine these targets? Do we do them individually? The way some of the thoughts are also, can you put 2 targets on the same CAR T? Do you have 2 different CAR T products and you give them together? Should we give them earlier in lines of therapy, or should we give them later because they have more toxicity? And I think each CAR T also has different side effects depending on what they're targeting, right? Like, so if it's BCMA, then there might be a little bit more immune suppression compared to if there's GPRC5D, which may have skin and nail side effects. So there's a lot of difference and individualization that's going to come. And I think we have a lot to learn from the bispecifics too, because once we start using the bispecifics, if that's going to be much easier to give. And I think the challenge may be a little bit more like the risk of infections long term. And so once we understand how that's working out in the real world, I think we'll be able to better understand. And the last thing I'll say is just access to CAR T. Just now, the companies haven't figured out how to make enough of it. So if that's not, done over time, then we'll just be using bispecifics because of that. So I think there's lots still to be determined, and the next few years will tell us.
And I wanted to piggyback on what was already said. We need better immune system. We need better T cells in a patient. There are attempts to get T cells from healthy donors, whole other kind of worms with challenges, because those T cells think, oh, wow, I'm in the wrong environment. I have to fight everything. But there are approaches like that so that the T cells are not harmed by the myeloma, as Dr. McCarthy said, or harmed by the treatments that we have done. And I think approaches to make those T cells stronger and better and longer living so that they don't die after a few months, but stay in the body longer and fight the myeloma longer, which is then more similar to the bispecific that we can give over months and hopefully years. I think that's another part. And then figuring out the side effects, why are they happening and what can we do to prevent them? I think those are future developments.
We're gonna think of your questions. I'm gonna come out and get your questions right after this, and we'll close this part with this question. My background is in politics and history. So as you can see, this morning was a lot of historical stuff. What is it what's the one issue or one problem you wish we could overcome soon? I mean, what's the one thing that you think is within grasp that we don't have yet? Or what do you see in the future that people should get?
You always have something to say. You got to think, all right,
Start with Dr. McCarthy.
medicine wise or economically?
Both. Any issue that you think these people should think about in the next year or two as they think of their therapy?
Well, they shouldn't have to think of the economics, okay? That's ours and politicians and insurance companies and all that. Although I do recommend there's a video by Katie Porter, and she just grills the head of makes venetoclax, AbbVie. And she gets out her little whiteboard and she talks about, and she goes through the fact that most pharmaceutical companies spend more money on advertising and taking out physicians to dinner than they do on R&D, even though they'll say otherwise. Oh, we have to have these large, I'm going to get very political, sorry. But so this is why you need to tell your Congress people and senators and everything else, look, we have to have a different approach to pharmaceuticals because the cost is ridiculous. She does the same thing to Mark Alice, who was then CEO of Celgene. Oh, the price of lenalidomide went up 5 times. Did it get any better? Was it more effective? And it still is that way. So that's number one. We have to have a different approach. This is a global across the board. We have to look at the cost of medications. And even when Len becomes generic, it's still going to be ridiculously expensive because we have to have the REMS program, which you're all familiar with. And I always love it. People in their 70s have to promise not to get their spouse pregnant. And so we really have to think of it differently because a lot of drugs cause mutagenesis, not just lenalidomide. So again, it's just because of the thalidomide thing. So anyways, that's number one. Number two is, yeah, we need better drugs. We need more effective drugs. We need to figure out how to use them better.
I think for me, the current major concern is the fragmentation of healthcare. If you get a prescription to a Walgreens, then every Walgreens in the United States knows the prescription. But CVS, they don't have a clue, and Walmart doesn't have a clue, and there's no sharing. And there are many patients who, for example, get their myeloma chemotherapy at one center, get orthopedic care at a second center, get bone building medicines at a third center, and see a cardiologist for an unrelated heart problem at a fourth center. And in some systems, there's a complete lack of communication between those four. And it's done by high-tech things like writing a letter to the other doctors. And given our system, the inability for multiple systems to share, so that if a patient is seen at Memorial and Roswell Park and at Mayo, it would be nice if I could instantly see what Dr. Shah, Dr. McCarthy had to say. That's not always the case. And with the way electronics have been developed, this fragmentation where different healthcare providers don't consistently have access, I think is a great disadvantage for a patient because their expectation, if you ask them, is that everybody knows what everyone else is doing, and that regrettably is far from the truth.
Just to add to that, I just heard recently about a company that kind of addresses that. I haven't used it too much with patients yet, but it's called Milo. And what they say is that a patient downloads the app on their phone and the doctor can send it to Milo. And then the patient can look at the prices at different pharmacies and get it sent to where they want to. So it doesn't have to, we don't send it directly to a pharmacy, but you send it to Milo, and then Milo, you pick the pharmacy you want. But that way, I think the cost comes, like, patients can choose where they're getting a better price, but also what's convenient, and not have to stick with one pharmacy the whole time. I haven't used it much yet, but I just heard of it. So I just thought I want to share it based on that.
So
think
with the Q and A, or somebody, okay, with the Q and A portion, we're having some mic difficulties. So I'm going to just put this in front of your mouth when you ask the question. But think about questions. This is really the roundtable part of our roundtables. This is where you're an equal at the table with everybody else up here. So think about the questions that are burning. They don't have to be sequential, and just what is burning in your mind, and based upon what you've heard today, what do you have questions about? We also have some online questions, and I'll get to those as we go along. But get to like like the auction thing. Just give me a little signal or something. No. We need it for the online audience.
All right.
I'll hold it.
Okay, this is a trivial question, but it should therefore have a quick answer. If one gets the Evusheld shot, which it seems as though all of us in this room should one also be getting the new booster?
The recommendation of 2 days ago from our infectious disease department is yes, keep 2 weeks in between. You should get both, because our treatments and your disease itself both limit your ability to build titers against vaccines, and the Evusheld overcomes that a little bit.
Yeah, just one caveat. You have to do vaccines first, then Evusheld. Because theoretically, Evusheld will block. I mean, your body will say, oh, I don't need to make antibodies. I got plenty against COVID-19, but it's only going to last for 6 months. So to Jens's point, Dr. Hillengass's point, you want to wait, like when if you got Evusheld, you want to wait till you're due to get it again, then get your vaccine, your booster, assuming you've been vaccinated before. And then you get the Evusheld afterwards 2 weeks later.
My 5th shot scheduled October 14th.
My 5th shot's tomorrow.
Thank you.
Yeah, us old people.
I've had all 5 shots and I had the Evusheld. When you say the Evusheld only lasts 6 months, does that mean we should get it again?
Yes,
Okay, schedule.
plain and simple. Again, the Emory study was very, very thought-provoking, because they show that myeloma patients just don't generate as high neutralizing antibodies to virus as other patient populations. We think that might be bad, but we don't know. That's the other problem, is you measure an antibody titer. I don't know what that means, because we don't have long-term data to show how protective something is. We're pretty confident about Evusheld, and this is for the patient population where you may just not mount a good response because you're on active therapy, or myeloma itself can cause immune paresis, where you don't mount good antibody or T-cell responses, both B-cell and T-cell responses to vaccines or pathogens when you encounter them.
Oh, wow.
So for the general public, they're saying that this new vaccine is like an annual vaccine, like a flu shot. But for us, we kind of were recommended to get boosters after 4 months, before. If we go and get this booster, which for me would be my 6th, are we still supposed to get another COVID vaccine like 4 months after, or is this a 1 year, one-time shot? I mean, I've had the Evusheld too.
We don't know. It changes every month.
He hasn't made any recommendations. So right now, I think it's 5, and then we wait. I mean, there have major people who are making the decision, so no one can answer the question. Fauci said it is feasible as he projects that we'd be getting an annual COVID flu shot. That's not solid. And so right now, I think the correct answer is 5 shots. And then we wait because we just don't know.
Now, of course, Rand Paul has other things to say about that when it comes to Fauci. Those are amazing interactions, but right now I can tell you that group at Boston Children's just published a paper, now it's in mice, showing that they may have found a common antigen to COVID-19 that will allow for, just like you had with polio, one and done, or you get a booster 10 years later. We don't have that yet. So right now it's like the influenza vaccine. It's like whack-a-mole because every year new variant comes up, wham, you got to have a vaccine. In fact, scientists spend all their time trying to anticipate which viral variants will be prevalent this coming year, and then they develop the vaccine for it. And some flu vaccines are better than others because they don't always anticipate which strains will arise. So I'm also getting my flu shot tomorrow. So, and my shingle shot. So we just don't know enough. So long answer to Dr. Gertz's point, we just have to wait and see.
You got a follow?
Yeah, just to follow up to that. So, I mean, I had my 5th shot 3 months ago, but now, you know, the guidelines say if someone had their last booster more than 2 months back to go and get this new one for the Omicron variant. Are we allowed a 6th shot if we are immune compromised right now, according to the guidelines?
How'd you get 5 until now?
Because 3 is the first, though, 3 was the first regimen and then 2 boosters.
It was actually 2 is the first,
Well,
then 2 boosters.
But then they changed it.
Yeah, get the Omicron. To Dr. Gers, you should get it, okay? Because it's not at all the same. I mean, it's a totally different variant than the previous ones. Plus, I mean, a lot depends if you got Pfizer, Moderna. There's some subtleties, but yeah, if you've not had the Omicron, I would strongly recommend you get it. The chances are you won't. If you weren't vaccinated now, you would have a high likelihood in ending up in the hospital very ill. And in fact, in our allogeneic transplant population, our death rate was about 50%. It was horrific pre-vaccine. That's why it makes me ill just to think about people who, for whatever reason, say, I don't want to get the vaccine, I'm waiting. One of our football players, Cole Beasley, said, I'm going to do my research before I get the vaccine. Like, what are you talking about? But I do say that you should get and keep up with the vaccines because it'll protect you so that even if you get COVID, you just won't get as sick. That's really the most important thing.
And just to verify that, Jenny had a CAR T transplant. She's on the CARtitude study and also had COVID. And because of her vaccines, it wasn't that big a deal. We have a question right here.
Actually, I've got 2 questions. First one, just with the CAR T, are patients only getting that if they're in a clinical trial, or is it something that you can try to get for a patient in your practice?
There are 2 products approved, but as we discussed earlier, the access to those is very limited right now because of the production issues. And they have certain criteria. And unfortunately, we have long wait lists where we basically try to keep patients alive and also in a good enough shape to get these CAR T cells. So at the moment, 2 are approved. There are studies going on. The studies sometimes have other inclusion criteria than most of the time have other inclusion criteria than the approved ones. So it's very specific to every patient, depending on if they're eligible for a trial, if they're eligible for the commercial product that would be paid by the insurance. Yeah, so there's all factors that we are considering.
Okay, and my other question is just clinical trials in general. I don't know much about them, but I know there's usually multiple arms. Are patients when they go into a trial, are they like the placebo concept? Are there patients that in a particular arm would not get anything, or does each arm provide some therapy for for each, all the patients in each arm?
So that's a good question. I think you're asking about generally clinical trials. And so scientifically, a trial cannot be done if one arm is so significantly inferior than the other, and it's going to cause problems to the patient. So it's usually a trial will have if it's a randomized study, each arm, like scientifically, we don't know that one arm is definitely better than the other, which is why the trial is happening. If we already know one arm is better, then that trial shouldn't happen theoretically. And so in the sense when if it is a placebo controlled trial, then we are thinking that placebo is the standard of care in that situation, and that's why it would be. But in myeloma, usually there aren't placebo controlled trials. They're usually with some other therapy in the other arm.
First of all, clinical trials are developed by global thought leaders. These are the smartest people in the world on myeloma as they sit down and meet on a regular basis to decide how to make things better for myeloma patients. The trials aren't developed to fail. The trials are designed to push the envelope forward and make things better for patients. That's one. Number two, the ethical regulations that underpin clinical trials and the federal oversight is staggering to ensure that everything is done ethically and appropriately. So for example, a trial of patient with previously treated multiple myeloma, there'd never be a placebo because there are effective therapies for previously treated myeloma. It's usually comparisons of different groups. Clinical trials also pre-specify the monitoring that a patient has to have. And I've seen what these clinical trials do, and the monitoring is better than what occurs in general practice. And so I think patients are actually being scrutinized in a much, much better fashion. So this whole concept of placebo and I'm a guinea pig is a complete misunderstanding of trials, and two, intrinsic distrust of the medical system, frankly, that your doctor's not trying to do the very best possible for you. So when the COVID vaccine was introduced and no people were ever treated with it, it was completely ethical. You didn't know if the vaccine was safe, and you didn't know if it would protect against COVID. So there was a placebo arm to determine if, but as soon as it was clear that COVID vaccine helped, all the people on placebo were unblinded, were told they received placebo, and offer the treatment. So placebo only occurs when there is no proof that anything other than placebo is any better. Dr. McCarthy did the very, very first trial after transplant of using lenalidomide, Revlimid, or placebo to determine whether treatment after maintenance treatment after transplant was helpful. He showed it worked. Countless lives have been saved since that time, and it's become the standard of care. But the opportunity, I'll bet Jenny's been in a half dozen clinical trials. I'd be shocked because she searches out the clinical trials and gets herself registered. And at least at our center, we have more CAR T capacity in clinical trial settings than we do with commercial CAR T. So one of the strategies to access some of the new technologies or the newest drugs is entering a clinical trial.
Yeah, I second everything Dr. Gertz said, because at the time we developed that trial post-transplant, that was 2001. There was no standard. I mean, there was thalidomide, but nobody could tolerate it for maintenance. And that trial opened up in 2004, and we didn't read it out for a few years later. It took a long time. But yeah, it was very ethical to have patients on placebo because we weren't even sure that lenalidomide was going to be better long-term. And as you saw the cautionary tale about venetoclax, that's why we do trials, because venetoclax looked very promising in smaller studies, but actually inferior overall survival. That's why we do placebo-controlled trials. And then lastly, my granddaughter, who was 18 months old, had the opportunity to participate in a trial at Strong. So she got randomized to either a COVID-19 vaccine, she was on Pfizer, or placebo. So she got unblinded 6 months later when she was like 2 years and change. She had gotten placebo. Then she got vaccinated. I had no problem with her being on that trial, even though she got a placebo, because it was important. To Dr. Gertz's point, we had to prove it works before you just do it.
Could you mention data safety monitoring?
Oh, yeah, yeah. There are such things as DSMBs, data safety monitoring boards. I participated in them as we all have. And what that is a group of investigators who are not part of the clinical trial, who have access to the data and see the data. It's usually done through a clinical research organization independent of the company, where we will go over data and then say, yeah, this is good, or "Ooh, we have a problem here." And so that's really important, is that there is, to Dr. Gertz's point, there's tons of regulations to make sure it stays safe for our patients.
I just wanted to ask if you could address patients with high-risk disease as it relates to these new CAR T and the bispecific treatments, and whether there has been any new research on that.
Not specifically for high risk. We obviously in high risk patients where we know, okay, there's a higher risk that the disease comes back earlier, that the treatments are not as effective. There's a medical need, as we call it, or an urgent medical need. To my knowledge, I mean, there are obviously, we do subgroup analysis, but the numbers of patients who have been treated with these new treatments are not very high, and you only have a certain percentage of those patients being high risk. So it's not clear if a patient with high risks responds better to CAR T, worse to CAR T. That's not clear. Unfortunately, what we have learned in history is that standard risk patients respond better to any kind of treatment than high risk patients. It's very rare that a certain drug works better in a high risk patient that it would work in a standard risk patient. I'm not aware of any drugs. I'm not sure if you have any.
Risk keeps changing. We used to have ISS. We had ISS 2. Now we've got the revised ISS. And revised ISS 1 and now revised ISS 2. Sorry, misspoke. And chromosome 13 abnormalities used to be high risk when Bart Barloghi first classified them. He really pushed the field. They're no longer. Once bortezomib came out, it no longer was high risk. So a lot depends on time and understanding. There's a paper I'm reviewing now. I'm not going to say what, but it's a large study, and they did a lot of molecular analyses, and they think they may be able to figure out which patients benefit the most from lenalidomide maintenance. Now it's an older study, but still, this is important. So the field keeps changing, and as we understand the biology of myeloma better, we're going to have a better idea of what is risk and what is truly high risk, and we may tailor things so that Dr. Hillengass's point, 11-14s may all need to get venetoclax early in their treatment because that may be the best for them long term. We don't know.
Hi, I just had a question about the CAR T. There has been a lot of discussion over it, and what piqued my interest was a couple of doctors. McCarthy, Gertz said it was, if I heard it right, overrated. And I was just curious if maybe we could talk a little bit about more of that and sort of organize those thoughts and maybe hear from Dr. Hillengass and Dr. Shaw.
You said overrated?
No, it's not the panacea everybody thought it would be. And it's the same thing with CAR T for lymphoma. You only have about 30 to 40% of patients who have long-term durable remissions and probable cures. That means the majority of patients don't benefit from that CAR. I mean, they benefit short-term, it may control their disease, but it comes back. And so that's why this isn't like the Kaplan-Meier curve remains at 80 to 90%. This isn't childhood acute lymphoblastic leukemia, where the long-term outcome is 80 to 90% cure. We're not there yet. That's why I think people were all excited that this is going to change everything. It has for certain patients, but not for everybody. And I think it's even more difficult for myeloma patients, because myeloma patients, for ide-cel, the median progression-free survival is about 10 to 12 months, which is okay. I mean, these are people with bad disease, but still, it's not the same. It doesn't even seem to be as close to lymphoma. CARTITUDE, the Janssen paradigm cilta-cel may be a little better, but there's no comparison yet. And to be frank, in our own experience, we've had patients with both ide-cel and cilta-cel who relapse fairly early, and we have a few patients who are doing quite well, and we just don't understand yet who they are.
There's not much to add. It's so individual, and we are still in the process of learning, as we do with everything. In the patients in this room who got a high-dose chemotherapy benefit from the experience that they made 20, 30, 40 years ago. And the patients in 30, 40 years will benefit from the experience that the patients now go through in clinical trials, and that's why we do clinical trials. It's not, we don't know enough to say this patient will benefit and has no side effects. It's just not possible right now.
Just maybe one last thing in relation to your question is that in the CAR T trials, patients who got the CAR T cells had to have been able to get chemotherapy, get the collection, have their disease under control, let the cells be made, which take about 4 to 6 weeks. Then they have to come back in for the infusion and all of these things. So they inherently had a disease biology that could wait for that time. So there is some, you see that in the CAR T studies, they've done better, but we're also selecting for patients who are able to do this when we do CAR T cells. In the real world, we are able to push that envelope a little more and say little sicker patients, little faster disease, higher disease burden, we're going to put them on the CAR T. So we probably won't see the outcomes as good as what we're seeing in the trials, though that is yet to be determined. But a lot of it in the CAR T world is this self-selection, whereas that selection is not there in other trials often, because you can put them on the trial immediately when they are ready to need the treatment. So that's something to think about with CAR T, which is not with other therapies.
I just want to distill, I think, what all the other speakers have said. CAR T will shrink the myeloma when nothing else is working. And so one can't poo-poo that. But again, distilling what Dr. McCarthy said, the myeloma with our current products comes back in half of the patients within a year. And that's, I think, what most people don't actually recognize.
Now, there are other products. So, for example, there are allogeneic CAR T cells in clinical trial right now. And that's where they're really interesting. I do recommend reading any biography of Jennifer Doudna or Charpentier, the two women who describe CRISPR, because CRISPR has allowed us all kinds of interesting ways to potentially genetically modify cells, including CAR T cells. So you can take any healthy person, you can knock out certain things that allow you to use the cells so it won't be rejected by the patient and won't cause something called graft versus host disease, where the CAR T would attack the patient. That would be bad as well. And then you put in a CAR, a chimeric antigen receptor against, say, BCMA, and that goes after the tumor. That's not ready for prime time. It's still in clinical trial. But that way you may get around to Dr. Shaw's point. You may be able to pull it out of the freezer and just use it for the patient. But the problem is it's still 101 because the majority of patients still progress. So we need to have a better understanding. Maybe we need dual CARs, GPRC5D and BCMA. Maybe we need to collect earlier. Maybe we need to do an allo or a CAR T and infuse it every month. These are all questions we just don't know yet.
With all of these medications and different cocktails, there is a potential for renal toxicity. What can we do as patients to offset that, manage that? What would be your recommendations?
Since it's very individual, it's also very difficult to answer this question. For a neuropathy, there are certain things we can do. For diarrhea, we can do certain things. for infection. A high risk is obviously the immunocompromised state of a patient on active treatment, because myeloma is a disease of the immune system, so there's collateral damage. So what we several times mentioned is vaccination is very important, being careful, using hygienic standards that are higher than for the general population. It depends very much on every single side effect, and it's very difficult to give a general answer to that. And you have to talk with your doctor, sitting in front of them saying, this is my side effect. And then your doctor can say, okay, this is what most likely will help you. And it's not even 100%, unfortunately, because one patient responds to a certain treatment and the neuropathy goes away. Another patient has no response whatsoever. We are very individual. Every single patient has a different set of response, side effects, duration of response. You can have a great response now, it can last very long, you can have a great response that you lose. So every single patient is different. That's what I meant with multiple myeloma. So every patient, and some patients have no side effects whatsoever, and that's great for them, but it doesn't help the patient who's sitting next to them in the waiting room who has a lot of side effects, right? And we try to learn more about it, and that's our job. But your job is basically to tell us what's going on. And I think it's also good to have things like HealthTree, where you can share your experience, because I have learned a lot, especially when it comes to side effects. I have learned almost most of what I do for my patients to improve their side effects from other patients who told me I have done that and it helped me, right? And that's a little bit try and error, but that's how we deal with that.
Most common cause of kidney damage in a myeloma patient is not the treatment of the myeloma, it's the myeloma. At presentation, 1 person in 6 with multiple myeloma will have impaired kidney function. By 6 months, 2/3 of those patients will have complete recovery of their kidney. And so you really have about 1 person in 20, 6 months into it, that has persistent abnormalities in kidney function. Actually, most of the treatments, with the exception of stem cell transplant, have very, very little impact and don't damage the kidney, although adjustment of dosing is required when the kidney is impaired, but they're not really very harmful to the kidney. Transplant, that can cause changes in the kidney, and that's why people focus just on doing transplant. So they have experience to understand what are the things that need to be done if a patient who has impaired kidney function decides to proceed with stem cell transplant. But there's a lot out there in terms of support and expert teams to manage that. But if you're talking about the conventional treatments for multiple myeloma, they generally don't damage your kidney. If the kidney's starting to go bad, you worry about the myeloma being more active.
We have some online questions as well. And remember, this roundtables are like one-room classrooms. So we have novices and experts in our classroom, and the novice questions are just as important. And I'm sure you'll answer this one quickly, but it's asked a lot. Is there a way to pre-screen for myeloma?
That's actually not a novice question, because there's a lot of research going into that. There's actually a study going on in the country of Iceland where they do screen for myeloma. The good thing is that this will take a long time to read out, because myeloma still remains a rare disease. MGUS is fairly common. The older we get, the higher is the chance that something goes wrong in our plasma cells and we start to produce a certain protein, which we call monoclonal protein. So this is actually a good question, but we still don't, at the moment, we don't screen because myeloma is fairly rare.
Okay. Second, another question about maintenance. And I'll condense this. So with respect to things like Velcade or Revlimid used in maintenance therapies 2, 5, 10 years, when is it the best time to stop maintenance therapy? And is MRD, is minimal residual disease measurement, or are other algorithms a part of that?
It'll take about an hour to answer.
Exactly.
Well, first of all, most of the studies that were done with bortezomib were for 2 years, sometimes fixed number of doses, almost always. There were no studies that were done continuous bortezomib because it's too toxic. So the large trials, again, were about 2 years, and there was efficacy, but the problem is the HOVON 65 GMMG4 trial. I said GMG4 because the Germans get always pissed off that we just say HOVON 65, which is Dutch. So that was comparing bortezomib to thalidomide. Well, that's stacking the deck right away because thalidomide's inferior. So that showed a benefit. And so then for a while, everyone's saying, oh, if you have deletion 17, you have to get bortezomib, not lenalidomide. But that's actually not true either. We have some other data that'll be coming out on it. Then, so what we don't know is when to stop because lenalidomide does have toxicity. Some people have low blood counts. The British and the Italians did 3 weeks on, 1 week off. In the US, it's every day. So there's a whole bunch of people who will often tolerate it better if they get a different schedule. So I think the biggest problem is we don't know, okay? And there are studies that are going to answer this question. For example, there is a SWOG trial. It's Dara-Len versus Len. And at 2 years in, you're going to get a bone marrow biopsy. And if you're MRD negative, you'll be randomized to stopping versus continuing. That's going to read out probably about 10 years from now. And I'll be really old. And then everyone would go, we don't do Lendara anymore because now we're doing something else. And that's the problem where we have to have surrogate endpoints for long-term outcome. And until we have those surrogate endpoints, we're going to struggle with what the right answer is because the trials take too long to read out. But there are studies ongoing to answer the questions that were brought up.
In terms of, Len discontinuation, there are 2 studies. I think there's one at MSK ongoing for lenalidomide or any maintenance cessation. And then there's one at the University of Chicago. These are small studies, but what they're looking at is if patients have achieved this MRD negativity or achieved a complete remission, and if they've been in this for a few years, is it okay to stop and monitor really closely? So I think we will see some of that data too coming out over the next few years. And that will also be helpful in making decisions around it, because now that we know that MRD negativity is important in terms of prognosis, and there are some patients on lenalidomide maintenance in MRD negativity for years, are those patients still needing the lenalidomide, or can we stop? And I think that's an important area that's being looked into.
Another question, is extramedullary disease considered high risk? And if you could explain briefly what extramedullary disease is.
Yeah, definitely. It is considered high risk. It's just harder to treat. That's part of it. And often as myeloma progresses, it tends to become more extramedullary or less secretory. So less of the myeloma protein in the blood and more in other sites. And so that makes it harder to treat. And one of the problems with that is that clinical trials often are looking at the secretory myeloma or looking at having an M spike. So as we get more extramedullary or non-secretory disease or disease that's oligosecretory or secreting less, then it's harder sometimes to get patients on trials, which hopefully will change as we design new studies.
You all know that myeloma is a cancer of the bone marrow. And from prior discussions, it's easiest to conceive as myeloma as weeds growing in your bone marrow garden. These weeds that grow in the garden need that garden soil to grow. In the laboratory, to try and grow myeloma, it's really, really hard because in the laboratory, you have dishes and plates, but you don't have that garden soil, that soil in the bone marrow. But myeloma sometimes can change. And when it changes in a more aggressive fashion, these weeds are capable of growing outside the soil. And so if myeloma appears in the lung or in the liver or in tissues that are not that bone marrow garden soil, that's referred to as extramedullary. The myeloma can survive outside the bone marrow. And historically, that really has been a big deal, that all of a sudden your myeloma grows outside your bone marrow, gee whiz. But like everything, what's good and what's bad depends on what treatment you have available. And as treatments begin to improve, things that historically were not good become easier and easier to treat. So whether extramedullary is good or bad is an always evolving concept based on how effective the treatments are. But in general, the principle of weeds growing outside of the soil and just popping up is generally considered different from the traditional weeds that only can grow within the garden and damage the overlying bone.
Editorial comment. I just posted a story today on our website. And it has a link to Dr. Gertz's, one of the versions that we have of Dr. Gertz's Weeds in the Garden talk. I highly encourage every one of you to watch it. We have 3 versions of it online. But you will, whether you're a novice or you think you know everything, you really should. Well, I say it's the equivalent in myeloma to like Beethoven's Ninth. It really is the best talk in myeloma. So I just happened to post it today, so I hope you all will access that. One more question about amyloidosis. The patient here is aware that there's more data and statistics on stem cell transplant and multiple myeloma. What's the latest on stem cell transplant with amyloidosis?
Amyloidosis is a complication that is seen in maybe 5% of myeloma patients, where the myeloma protein actually deposits into the tissues and causes organ damage. For the majority of people here, it's irrelevant. It's not happening. This is a very, very uncommon problem. 1/5, 1/8 as common as myeloma. But the standard of care currently, by consensus, by agreement among specialists, is that you should get chemotherapy as defined. It's chemotherapy. And currently, there's a 60% chance all the amyloid proteins will go away. And if that happens, nothing further needs to be done. But for patients who don't have it all go away, or at least go away 90%, and if transplant can be done safely, the safety profile is very different than myeloma transplant. And there are many patients who have amyloid where it would be unsafe to do a transplant, so it isn't done. But if you get chemo and you really haven't had a big, deep improvement in getting rid of the myeloma proteins, then stem cell transplant is done next in most centers for when it can be done safely, to try and drop those proteins down to the lowest possible level. So it's used, but the frequency with which it's used in the last 3 years, it really has plummeted significantly.
Completely agree.